Hi, good evening. On this series, we'll be looking at using the Flowjo. Many of us would want to run analysis using the flow cytometric means to detect cells, all kinds of cells, most especially the immunological cells, to see the response of some uh, response how the immune system will respond to foreign antigens, and also possibly to look at markers, immunological markers, and also the response of cancer cells. Anybody who's working on cell lines will most definitely use the flow cytometric analysis. Now, the flow cytometric analysis kind of is dynamic, very dynamic to accommodate all forms of cells, be it cell and whatever cell it is, it will be able to measure it. One thing about the flow cytometric analysis here, when you look at the hydrodynamic focusing, when it passes through the sheet fluid, and to measure it from between the long pass and the short pass, and you have whatever it is measured in frequencies. And with these frequencies, you can further do the analysis using the Flowjo. Here before us is the Flowjo software, and we have by it we have um, analysis from the flow cytometry. It's one of my analysis. So I'll just give you a little rundown today on what is expected and how the Flowjo looks like. Typically, this is the background of the Flowjo. You're gonna see it look like this, and it tells you drag sample here, and we have the sample on the right side of my screen. So I'm going to lift because we have in the flow cytometric analysis, you have monoclonal antibodies or polyclonal antibodies, and which you have used to stimulate the cells, and then you have beads we use in marking these cells so that we can measure their observance through the flow cytometric analysis. Not just the observance, you're looking at the size, the shape through the fold, and the side scatter, the granularity and all that. So we have the compass beads, you have the FitC compass beads, you have the PEC24 compass beads, you have the PE Texas red compass beads, and all the compass beads that we are going to use based on the observance. So now this we in this analysis we have three compass beads. We have the PE CD4 compass beads, we have the APC CD200 compass beads, and we have the FITC 200 FITC 200 receptor compass bead, and we have the single non-staining control. So I'm going to pick highlight, and then this is just simple how it's done. You lift with your cursor and you drop where it says drag sample. I have done that. And the next process we're going to go about is trying to assess it. The process of where we in everything you have the living, the cells that are alive, and those are apoptotic cells, and those ones are alive. Basically, what we are looking for is we're not looking for the apoptotic cells or debris. We are looking for um, the lymphocytes. So the next step we're going to do is that we're going to get the lymphocytes. In When we talk about the beads, you have different ratio. You can see the screen is blue, and you're wondering what this is. This typically it makes reference to the beads. This particular one is the beads, the color of the beads. From here onwards, what you are looking at from here down here, these are all apoptotic cells, dead cells, or debris you find here. And reaching out here up, as you move up outside, these are cells that have high granularity. From here you have cells that have that are larger in size and all that. And so as you can see, what we have here is side scatter, and what we have here is forward scatter. So I'm going to gating. I want to get the variable lymphocytes that we have. From there, we'll be able to get out CD4. So I'm gating, and then we are going to call these variable lymphocytes. Lymphocytes. That is to say, these are the lymphocytes that are alive. And in the gating, I went too far. There are several of courses we can use to gate. We can use the the square, the cross, the circle, the polygon to gate. So now that we have gated, to be able to get the others, I'm going to lift with my cursor and drop here in all samples. So if you look at it, it's fine that it's gated all of them. So we can quietly look inside and see what it looks like. What is trying to tell us that everything from here are variable lymphocytes. Everything here, variable lymphocytes. And you can see the frequency here, 2491, and all these are the frequencies of the variable lymphocytes and your single staining non-controlled. Now, the next thing we are going to do is we're going to create a layout. This layout is to enable us gate. We want to gate. In gating, we are going to put them in their various quadrants. So according to our template, what we are trying to look out for CD200 and its receptors. So what we have is your CD44 compass, CD44 compass beads. We have your CD 200 feed C, and we have your APC CD200. So in the first protocol we have is feed C, to your compass bead A. So this protocol, now that we have this, we are going to try to replicate this on the layout. So I go back and quietly lift and drop on the layout. But we know that we have three, the protocol is actually three outside the non-staining control. So we're going to try to duplicate this. So in duplicating, we just tap your cursor and duplicate like this. And there we go. We have, but now we have duplicated because not everything looks the same. So we are still going to change the protocol. So this is APC. Yeah, here we go. Okay, so at this we are going to try to gating in the various quadrants. So we are going to use this. So we are going to measure where the line is, where it meets between the two of them, and so we drop. Now you can see the quadrant with its percentage showing clearly. What is equally trying to tell you is that it has gated and it's telling you precisely where the lymphocytes are. So like we said, these are apoptotic cells, and it has been fully compensated, which we normally do with the flow cytometric and the flow cytometer. So everything from here now is saying that these are the variable lymphocytes. As you can see, it's telling you it's about 43.8%. So what I'm going to do is that I'm going to gate for all. Oh, we skipped a step. My apologies. So okay, let's just go back to step B. It actually caused no harm. So I'm going to highlight and organize them vertically, and then come back. I'm going to batch them according to the protocol. So that's batching. Now you see all of them batched according to the protocol. So I'm going to put them vertically also. like this. All right. And then we're going back to the Flowjo itself. So in doing this, we then lift what we have gated and we put it in the variable lymphocyte like this, and it gates for all of them. So we move to the next protocol. And we gate. As much as we try to be economical, and we shouldn't learn not to miss the line. Now that we've gated, we're going to do the same thing, and we lift and we drop. Now, if you look at our layout, to find that we have succeeded in gating all the protocols that we have, all of them, just left with the last one, and we are going to do that now. Then proceed to gate for your CD4 cells. So we'll go to the second protocol, which is your CD200 fit C, the receptor itself. So that's the protocol, fit C, compass bead, APC. Okay. And we do the same thing, and we drop. Congratulations, you have succeeded in doing that. If you're following, to get the Flowjo is quite easy. You can go online to download the Flowjo. You have the trial version, and you can also pay for it once you have your statistical analysis. There's really no need for you to hand over analyzing your Flowjo to someone else. You can typically do it yourself, looking at these cells when you have analyzed your samples using the flow cytometric analysis. The flow cytometer is very dynamic, gives you almost anything accurate once you're dealing with cell lines, whatever cells, whatever culture cells that you have run, immunological cells, whatever it is, the flow cytometer can analyze it. So now that we have gated, let us now gate for the CD4 cells. So in the variable lymphocyte, like we said, remember we said that the PE Texas is the CD44 cells where we have the CD4. So we put here the C Texas, and what we have next is for your forward scatter. Remember we said that the forward scatter will tell you the shape. You know that the CD4 cells are usually bigger, while those your from the myeloids, or the myeloid wing where you have your macrophages, and here that you have they are kind of granular. So now everything we have at the top here is your CD4 cells. So I'm going to gate now like this. Careful not to pick some of the apoptotic cells. So we are going to call this fellow CD4 positive cells. From here, we can go ahead to run statistical analysis if we want. For example, you can run the mean of your CD4 cells, and that we have done. You can also do the same thing for your APC, your CD200, and your CD200 receptor. But now we've done this, we are going to gate, lift and place, and do the same thing for the other three. That now applies to every other cell. So the next thing we are going to do in the CD4 cells, we are now going to gate for the CD200. Congratulations, we have gated for the CD200 if you're following. And so we are going to drop same thing. And then if you look at it, it's giving you the percentage of the CD200 in your PE Texas. And when you scroll down, the same thing to CD245. And okay, okay. So my apologies, we'll go back. There's something we missed. Actually, in gating, you gate it in the CD4 cells. So it runs through all of them now. Okay. Okay, that's for that. So now we are going to add our samples to the protocol. We'll come back to show you how to do the contour, but let's just analyze one of our samples and we'll look at the percentage of CD200 and its receptors, and also your CD4 in the biopsy mouse. And what we did was we got the lymph nodes and the spleen from these rats, both from the memory and the naive. That's the rats, the naive rats who have not been exposed, and the memory we challenged them with CD3, CD28, CD3 and 28, CD3, 28, and CD200, the blocker, the receptor, to see how they express themselves during stimulation from a foreign antigen. It is believed that CD200 itself has an immunological role it plays, an inhibitory or suppressive regulatory role it plays for the cells to prevent the cells from going through exhaustion, or where there is prolonged pro-inflammation or inflammation in the cells. The CD200 and its receptors play a vital role, but now it's been documented in rats. So we are trying to see if the same thing applies in humans. We missed something, so we are going to add that now. That's why I said the flow cytometer is the Flowjo is quite very easy to handle. When something is amiss, you can tell quite easily, and what you have to do is you follow up and you update. we're gonna get it again. So I call this your CD200. Okay, like we did before. Okay, I've done that. So let's add samples to the protocol now. Gonna drop our sample somewhere here and let it run. So we are still waiting on the sample. Once it's gone, we'll look at just one analysis before we call it a day. We'll come back again to show you how we gate forward the cells and what else we do with the Flowjo and how we can extract information from the Flowjo to Excel. In another analysis, we'll be looking at how to use Excel to analyze your ELISA reports, to draw your concentration curve with ELISA. And then we'll also look at your qPCR, how to analyze data with your qPCR on Excel. I'm sorry I didn't initially introduce myself. I'm Nana Isaiah Ibe. I'm bringing you this training so you could do it yourself. No need paying anybody money to analyze your statistics or your information as a researcher. All you need is a very good laptop, and these softwares, many of them are pretty available online. You can get them, and once you have them, you run your analysis. Yeah, they are popping up. They're popping up. Slow, but they're popping up. Yeah, they're all up now. They're all up, popping up. Yeah, so now we have them. So what he's trying to tell you that in the ones that we block the CD200 receptor, actually the CD200, since it's blocked, all you have is 3.14%. And this is for your memory cells, the ones that we have initially sensitized. And for your CD2, CD200, your CD3, CD28, the ones that we sensitized, still saying the same thing. Your CD4 cells fully expressed 84.5%. And then if you look at your CD200 in the CD3 cells, you have is 2.5% also. And then in the CD28, that is 0.98. So whatever role the CD200 performs, we would find out. That's for the naive. Now we are looking at the naive. For the naive, you have is 7.43. You have um, for your naive CD328 is 18.3. For only your CD3 which acts as an antigen for your CD4 cells on the cell on the surface of the cells at the receptor, you have it at 6.41. And for the one that is not actually your CD28 is 5.7. All these are for your the naive cells, and the analysis was done in triplicates. Um, we'll call it a day today. Next time, I'll show you how to run a contour. Everything here you see is actually your CD4 cells. And so, since we know that everything here is your CD4 cells, we can actually do a contour map to see what it looks like. So, um, I would try to do more analysis on this so you can actually do it yourself and be comfortable while you run the analysis on your own without fear or favor, without bias, without wondering what the person who you have handled your sample from, what he's doing with it. So for those who are interested, if you're interested in knowing more, you can reach out to me and we'll discuss on how and where you can get more informations on this. Science, medical science is dynamic, and as we continue to advance into the system where we have more dynamic methods of deciphering or analyzing specimens for accuracy, precision, reproducibility, and specificity, more of these softwares will continue to come your way. Thank you so much. Once more, my name is Nana Isaiah Ibe.